Okay, now tissue culture. Okay, we have root and shoot development. Okay, helped by auxins, the presence of auxins and cytokinins added to the medium that the plant is grown with, that allows for the differentiation into roots or buds and stems. Okay, so here's a root. Okay, here's a shoot with a leaf and a stem. Here's some flowers. Okay. Now plants, asexual or sexual reproduction. So plants can do both asexual and sexual reproduction. Okay. Now sexual reproduction, remember we talked about asexual reproduction is when a plant reproduces from root or stem or leaf. Now sexual reproduction has to do with sex cells or gametes or sperm and egg, and have to do with a flower in flowering plants. Okay. So generally, in meiosis results in gametes with half the chromosome number of the parent cell. Okay, so generally all cells have two sets of chromosomes. Okay, one set from mom, one set from dad. Okay, one set from mom, one set from dad. And so one chromosome from dad might be matching having the same genes and being the same size as the one chromosome from mom. So these are called homologous chromosomes. Okay, they're the same size, they have the same genes at the same locations. Okay, they're the same size, same amount of DNA, they have the same genes. Okay, so what would happen if the gamete chromosome numbers were not reduced? Okay, think about that. I'll let you think about that, and you can try to answer that question. Okay? So sexual reproduction allows for new gene combinations. Okay? So crossing over at prophase 1 recombines maternal and paternal genes. Okay? Segregation at anaphase 1 is randomly assorts maternal and paternal chromosomes. It's another way that you get new gene combinations. Okay, it's kind of showing you crossing over. Okay, here's one part of the chromosome and another part. So here's parts of chromosomes from one and parts of chromosomes from another, actually crossing over. Okay, so this is one chromosome, this is another chromosome, and here's a recombined chromosome with genetic material from mom and from dad, and from dad and from mom. Okay, so the crossing over can happen during prophase 1 of meiosis 1. So new gene combinations also result from fertilization, okay, because you have the sperm from dad and the egg from mom coming together and uniting, forming a zygote with a unique combination of genes. So all organisms that come from sexual reproduction are all genetically varied, okay? So during meiosis, DNA doubles prior to meiosis. Now, when does that happen? Think about that. Okay? Think about that, and you can try to answer that question. When does DNA double? Okay? See, during meiosis 1, reduction division, it's called sometimes. You have prophase 1, where chromosomes condense. Homologous chromosomes pair up. Okay, the nuclear membrane breaks up or breaks down, and crossing over can occur between homologous chromosomes. That allows for the exchange of DNA, like we saw in that slide a couple of slides ago. Okay, so prophase 1 allows for recombination, and that's a source of variability. Okay, another thing that happens during prophase 1 is that the spindle forms. Okay, now in metaphase 1, the homologs would be aligned at the equator of the cell. Okay, and the spindle becomes more apparent. You can kind of see that a little here, right here, the spindle, this slide, or this photograph. Okay, in anaphase 1, the homologs migrate to opposite poles, as you see here. with the spindle pulling them, the spindle fibers pulling them to opposite sides of the poles. Until the phase 1 is complete when one cell becomes 2. So after meiosis 1, it's called reduction division because after meiosis 1, the chromosome numbers have been reduced by half, right? One set of chromosomes went to one cell, another set of chromosomes went into the other cell, okay, of the homologs, right? So why do plants have sex? Okay, you could have a population on a mountain in Hawaii, and they need to actually propagate themselves and survive. So of course you'll have hybridization between one plant and another plant, okay? Or even a plant could self. That's a form of sexual reproduction too, okay? Another reason why plants have sex is that environment can change. It can get hotter or it can get colder. There could be more or less water available. There could be more fog or more pollution in the environment. So if the environment changes, the plants may go extinct, okay? So plants may go extinct if the environment changes, like we have global warming happening right now, right? So sexual reproduction is a key. It allows for new combinations of genes from two parents, okay? So meiosis kind of helps you reshuffle the gene combinations, okay? So what you have is meiosis. So you have different kind of chromosomes actually allowing for fertilization. And so when fertilization happens, you have chromosomes from two different sources coming together after meiosis have happened to the two different sources, okay? So that allows for new combinations of genes from two parents, okay? And also remember that meiosis reshuffles the gene combinations, okay? So the crossing over sites are not random. They're actually recombination hotspots, or places where recombination are 70 times more likely to recombine at certain places on the chromosome. Okay? So the areas near the centromeres are reduced, crossing over areas. Okay, so they think maybe that's actually that genes are not randomly placed on chromosomes. It may be important genes tend to be closer to the centromere. Okay, so here's a look at locations of genes. So here's a centromere. So these genes here might be actually the more important genes, okay, on the chromosome, just because that's less likely to have crossing over happen there, okay? So our housekeeping genes near centromeres, okay? And are genes allowing adaptations to environment in hot spots? Probably, okay? Now the alternation of generations, we have ploidy, okay? That means that you have different sets of chromosome numbers, okay? If you have one set of chromosomes, you have the haploid number, or X, okay? If you have two sets of chromosomes, like we do in our body cells, we have the diploid number, or 2X, okay? So here's a diploid cell with two sets of chromosomes. Here's a haploid cell with one set of chromosomes. Now if you have three sets of chromosomes, you can have triploids, okay? And that happens in plants. Okay, that doesn't happen in humans, but it happens in plants, okay? So some examples of triploid plants include navel oranges, seedless watermelons, okay? And that's one of the reasons why they don't have seeds, okay? And then you have four sets of chromosomes, which are called tetraploids, right? And tetraploid plants include potatoes and pasta weeds, okay? Now, for most animals, almost all animals, the gametes are the only haploid cells. Remember, gametes are the egg and the sperm, right? So for most animals, only their eggs or their sperm are haploids, okay, or haploid cells, okay? But in plants, you have the alternation of generations, okay? What you have is you have a 2X sporophyte in the diploid phase, okay, alternating with an X gametophyte in the N phase, okay? So the sporophyte is a plant that produces the spores. The gametophyte is a plant that produces the gametes. Okay? Here you have the gametophyte giving rise to gametes that come together to form the zygote. And here you have the adult sporophyte here, undergoes meiosis to produce the spores, and then and that actually grows into the gametophytes. Okay, so the cycle repeats. So in plants you have alternation of generations. Okay, also in some protistans or protists, in fungi and in plants are the alternation of generations happening. Okay. Now there are 6 rules for alternation of generations. The first rule is that the first cell of the gametophyte is a spore. Okay, the last cell of the gametophyte is a gamete. Number 2, the cells of the gametophyte are haploid. 3, the first cell of the sporophyte is a zygote. Okay, the last cell is a sporocyte. Okay, and 4, cells of sporophytes are diploid, okay, or 2N. Okay. Now 5, the change from sporophyte to gametophyte occurs from meiosis. And lastly, number 6, change from gametophyte to sporophyte occurs from fertilization. So we see here fertilization, then you have the diploid, zygote, sporophyte, then you have meiosis happening, producing spores. It gives rise to the gametophyte that produces gametes that come together in fertilization. Okay, that results in the zygote, and that alternation of generation continues. Okay. Now back to sexual reproduction. Okay, it allows for new gene combinations. All right, you have the crossing over at prophase 1. Recombines maternal and paternal genes, okay, and segregation at anaphase 1 randomly assorts maternal and paternal chromosomes. That sounds kind of familiar to me. I think we kind of went through this, okay. Anyway, so Barbara McClintock, okay, she worked at Cold Spring Harbor in the 1940s and the 1950s, okay, and she worked alone pretty much, you know, away from all the mating crowd, sort of. And she discovered these things she called transposable elements or transposons. They're called jumping genes in corn. So 85% of the DNA in corn is from jumping genes. Okay? Now molecular genetics. Okay? Chromosomes have to do with chromosomes, and chromosomes are made of DNA and protein. Okay? Now the molecule of genetics is DNA, deoxyribonucleic acid. Okay? And of course the units of deoxyribonucleic acid are nucleotides. Okay? They include a sugar, a deoxyribo sugar, a phosphate group, okay, and a nitrogenous base. Okay? So the nitrogenous bases of DNA include the purines, which are adenine and guanine, and the pyrimidines, which are cytosine and thymine. Okay? So you have a sugar phosphate backbone that actually forms the outer part. You have sugar phosphate alternating along the rungs of this DNA stepladder. Okay? Sugar phosphate backbone. And then you have the bases that are hydrogen bonded. So here again we see the importance of the hydrogen bond. Bonding the bases A pairing with T, as you see here, and G pairing with C, as you see here. Okay? Now Watson and Crick were the people who actually discovered the structure of DNA. Okay? There was a race to actually discover the structure of DNA, and it was actually helped along by Linus Pauling, even though I heard that because Linus Pauling was considered an unfriendly person. I mean, he's American. He was American. He worked at Caltech, but he was considered to be an undesirable, okay, because he had sympathies leaning toward the left at the time. So at that time, he was not allowed to travel scientific conferences. So some people actually say that Linus Pauling might have been the person to discover the structure of DNA if they had not imposed limitations on his travel and limitations on his work. Okay. Now Rosalind Franklin did X-ray crystallography. Okay, and that actually has to do with isolating a substance and then crystallizing it very painstakingly, hitting it with X-rays and forming a crystallograph. Okay, and Aaron Chargraph actually lent his expertise to it by finding out that the number of purines approximately equaled the number of pyrimidines. Okay, so there's Linus Pauling with his alpha helix. Here's Rosalind Franklin with X-ray crystallography, and Erwin Chargraph with his purines equaling the number of pyrimidines. Okay, so they actually helped Watson and Crick lead to their discovery of the double helix of DNA. Okay. Now DNA functions, what does it do? It stores genetic information, and most of this is in the nucleus. Most of the DNA is in the nucleus. Okay? So the information is in the sequence of nucleotides. Okay? So a gene segment is a segment along a chromosome that encodes a protein. Okay? So the genome is all the DNA in the organism's chromosomes. Okay? So for complex eukaryotes, you could have 10,000 to 50,000 genes. Okay? So bacteria versus plants, many differences between them. But corn versus teosinte, even though they don't look that similar, okay, they actually are very similar to each other. So they think that maybe corn may be descended from teosinte. Okay? So here's a look at a cell. There's a nucleus right here. Okay, with the nucleolus. Now, gene expression. So each cell has the same information. Okay. So different subsets are read. Okay, so epidermal cells actually produce cuticle, whereas mesophyll cells produce chlorophyll, okay, in a leaf, right? You would not have an epidermal cell producing chlorophyll, and you would not have a mesophyll cell producing cuticle, okay, just like in our bodies, right? Okay. See, here are epidermal cells, here are mesophyll cells. Excuse me. So DNA also stores genetic information, most of it in the nucleus. So replication happens when? Okay, so DNA is unzipped by DNA polymerase, right? Each strand is a template for a new strand. That's what they call semi-conservative replication. Okay, so the nucleus has enzymes that help to proofread DNA, but there's about 1 in every million bases that are incorrect. Okay, that just happens naturally, happens in our cells as well as happening in plant cells. About 1 in a million is incorrect. Okay. Now we also have RNA, okay? So transcription and translation, these are parts of protein synthesis, okay? Transcription happens when DNA codes for RNA or ribonucleic acid, especially messenger RNA. So there are differences from DNA and RNA. For one thing, RNA is shorter, DNA is longer. RNA is single-stranded, DNA tends to be double-stranded. RNA has a different sugar called ribose sugar, okay, whereas DNA has a deoxyribose sugar. And RNA has adenine pairing with uracil and cytosine pairing with guanine, whereas DNA has adenine pairing with thymine and cytosine pairing with guanine. Now, transcription results in messenger RNA, also known as mRNA. Okay, it carries a message, okay, from the DNA template to make proteins. We also have transfer RNA, which is also called tRNA. Transfer RNA has a amino acid binding site on one side and has the anticodon on the other side, okay? Because the DNA code is read on the messenger RNA in codons. Those are triplets of bases called codons, and the anticodon that matches up with the codon is on the transfer RNA, okay? Thirdly, we have the ribosomal RNA, or rRNA, which is a component of the ribosome, okay, along with proteins. Remember, the ribosome has a small subunit and a large subunit. Okay, so generally the start codon starts tRNA with methionine and the anticodon for that. Okay, that's actually translation in the cytoplasm. So UAC, the anticodon pairs with AUG. So AUG is the start codon, and UAC is the anticodon on the tRNA. Okay, so and joins the ribosome. Okay, so here you have the anticodons on the tRNA, the codons on the messenger RNA, and this is actually the ribosome. Okay, so what happens? You have the movement of the ribosome along the messenger RNA or the mRNA. Okay, and this is actually showing translation. Okay. Next tRNA pairs with the next codon, or the next tRNA pairs its anticodon with the next codon, attaches its amino acid in a peptide bond. Okay, so you see the peptide bond is going to occur between these amino acids, and then the tRNA goes away, and then the thing moves over, and then another thing happens. Okay. So the genetic code is read in triplets of nucleotides called codons, okay? And the order of the codons equals the order of amino acids. One of the things that's important to remember is that the genetic code is universal, okay? Genetic code is universal. Everything from bacteria all the way to the highest level of organisms have the same genetic code, okay? So the tRNA is like a translator. You have the anticodon on one side and the amino acid binding site on the other. Okay? So the flow of genetic information goes from DNA to RNA to protein, or to the polypeptide. Okay? So DNA codes for RNA in transcription, and RNA codes for protein in translation. Now, mutation, what is that? Those are changes in DNA sequence, and it happens all the time, like I indicated a little earlier. Okay, so mutagens also cause mutations. Okay, so mutagens include UV light, ionizing radiation, different kind of chemicals. So DNA repair often corrects mistakes. So DNA polymerase oftentimes corrects mistakes. Okay. Sometimes you have a somatic mutation which happens in the body cell, so that can result in a sport. So that resulted in navel oranges, the triploid navel oranges. Okay, also happened in red delicious apples. Okay, now germline mutations happens in cells that produce gametes. Okay, and is passed on through future generations. So that goes to variability. So that can include variability in flower color and smell. These are actually mutations that can result in different kinds of variability, such as flower color and smell, a taste of fruit and its texture, yield of grain and the quality of the grain, and disease and stress tolerance. Okay, so the accumulation of mutation, okay, actually leads to what? What does it lead to? You can have an accumulation of mutations that leads to new varieties, leads to new cultivars. Okay. Now, monohybrid crossing, studying a single gene. Okay, a true breeding parental generation differs in one trait and produces an F1 generation. When you intercross the F1 generation with each other, the first filial generation, it produces the F2, or the second filial generation. Okay, filial means son. Okay, so if you had a homozygous green potted plant crossed with a homozygous yellow potted plant, all their offspring had green pods, okay, then you know that green is dominant and yellow, or lowercase g, are recessive, okay? So all the F1 will have big G, little g genotype, okay? All offspring are heterozygous, okay? Now when you cross this F1, if you cross it or you self it, so you have big G, little g times big G, little g, actually shown here in this Punnett square. What you'll get is 1 big G to 2 big G, little g's to 1 little g. Okay, so what does that tell you in terms of what they'll look like? If there's a large letter or an uppercase letter present, you know that it's actually going to look like the dominant traits. You know that you'll have 3 green potted fruits bearing plants to 1 yellow potted fruit bearing plant in the F2, okay, or 3-to-1 ratio. Okay. Now, crop plant evolution. Remember we said earlier that there are approximately, or there are more than 250,000 species of angiosperms, and 6 to 11 species provide 80% of the calories for humanity, and they include wheat, rice, corn, potato, sweet potato, and cassava. Okay, so let's repeat that. Wheat, rice, corn, potato, sweet potato, and cassava. Okay, provide about 80% of the calories for humanity. Okay, and 8 species complete the list of major crops: sugarcane, sugar beet, bean, soybean, barley, sorghum, coconut, and banana. Okay, these are the major crops in the world that feed all of humanity. Okay, I'll let you look at that for a second. Okay, now the origins of agriculture. Okay, so the first humans were hunter-gatherers, that's what we think. Okay, maybe did they have better lives than agriculturalists? Okay. For one thing, hunter-gatherers have a better diet and they have better health. Okay, they're more fit than agriculturists. Okay, they eat more varied foods. Okay. They also have a lower starvation rate than agriculturists do. Okay. They also work fewer hours. Okay. So can we say maybe that hunter-gatherers have better lives than agriculturists? Kind of leave it up to you to answer that question. Okay. Now, gene splicing in transgenic plants. So we have what we call recombinant DNA technology, because DNA behaves the same way because the genetic code is universal, we said a few minutes ago, right? So we have restriction enzymes that are isolated from bacteria at first, okay? So what they do is they cut DNA at specific sites and they leave these overlapping pieces called sticky ends, okay? And these sticky ends will base pair with other DNA cut with the same enzyme, okay? That's the way they do gene splicing, okay, and make transgenic plants. So when you clone the gene, you can clone the gene that way, okay, with bacteria. So you actually can have a bacterial insect-resistant gene, okay? So you can have a insect-resistant gene put into a bacterium, okay? So first you need to find the gene, cut it with a restriction enzyme, then cut a plasmid, which is some bacterial DNA that's separate from the bacterial chromosome. So you cut the plasmid with the same restriction enzyme, then you mix them. And the bacteria will multiply and replicate their recombinant plasmids, and so the bacteria clone the gene. Okay? And you can have millions of copies. Okay, and you can insert them into plant cells. Okay, and then we remove gene with a restriction enzyme and insert into Agrobacterium tumefaciens. This is another way you can actually do transgenic plant. It's a bacterium. Okay, and infect plant and inserts DNA. Okay, so that's a couple of ways you could do it. Okay, you can also use a particle gun. Okay, it shoots DNA into plant tissue, and the pellets are coated with DNA. Okay, so that has resulted in transgenic corn, soybeans, cotton, and canola. Okay, it has actually allowed for disease resistance, insect resistance, and herbicide resistance. Okay, and it also leads to bioreactors to create pharmaceuticals. Okay, that can actually result in vaccines for hepatitis B, for rabies, for cholera, tuberculosis, malaria, diarrhea, cavities even. Okay. You can make biodegradable polymers. You can sequester heavy metals to actually bioremediate pollution. Okay. We can actually have ornamentals that result from gene splicing in transgenic plants or genetic manipulation, such as blue carnations, mums, and roses. You can extend the shelf life of cut flowers. You can have compact plants. Fragrance genes can be added to certain flowers to make them fragrant. Okay. So there are more than 395 million acres of transgenic plants grown worldwide. So in the United States, 93% of the cotton, which is also a very heavy feeding, high maintenance plant, okay, 86% of the corn, 93% of the soybeans, and 95% of the sugar beets, okay, are transgenic plants, okay. Also soybean, corn, cotton, and canola. Okay. Okay, so you have a gene that actually lends a plant resistance to Roundup, okay, which is a herbicide, broad spectrum herbicide. Okay, now another topic, we have micropropagation for sterile conditions. Okay, so that can result in disease-free plants. So micropropagation or tissue culture occurs under sterile conditions, okay, for disease-free plants. You can actually grow a large number of plants in a very small area, like just one room. You can have thousands of plants, okay. You can have rapid multiplication, okay. It's similar to growing cuttings. It's just another form of asexual reproduction. Okay, there are 4 steps in tissue culture. First of all, you do tissue culture. Okay, so you disinfest the plant first with bleach or ethanol, okay, in order to make sure that it's clean. Then you insert the plant tissue into a test tube, okay, and using a laminar flow hood, which kind of it actually pushes air out the front of the hood, so that bacteria are not flying in to the area where you're doing your tissue culture. Then you get sterile, viable plant tissue cultures. Okay. Now changing the subject. Okay, we have the early development of evolutionary thought. Okay, so more than 2,300 years ago, Aristotle thought that organisms are static, that they do not change. Okay, so fossils had been found for centuries, but not identified as such until the 1500s, okay, by Leonardo da Vinci, the genius. Okay, that he figured out that there are parts of organisms that were extinct. So there's Aristotle, and there's Leonardo da Vinci. Okay, then in the 1700s, Comte de Buffon, Georges Louis Leclerc de Buffon, okay, had evidence that organisms changed. Now in the 1700s and 1800s, they used comparative anatomy to classify animals. And in the 1800s, Jean-Baptiste Lamarck actually thought that changes in populations happened over time due to acquired traits, the inheritance of acquired traits, like the neck of the giraffe getting longer as it reached for leaves that were higher in the tree. And then what it did is that when it reproduced, it passed on its longer neck to its offspring. Okay, so it would reach up its neck and then pass on this longer neck onto its offspring. So that was changes in population over time due to acquired traits. Okay, so a giraffe's long neck. Now, Charles Darwin, in 1859, he published On the Origin of Species by Means of Natural Selection. Okay, that organisms with favorable characteristics tend to survive and reproduce. Okay, and this is the book, one of the most influential books of all time. Charles Darwin had been voted the most influential person of the last millennium. Okay, so organic evolution, okay, is accumulated genetic changes in populations over generations. Okay, so the first revolution was in 1831. Darwin set sail on the HMS Beagle. Okay, he was a ship's naturalist. He always had a love for nature ever since he was a little boy. Okay, he wrote about his discoveries, and after about 20 years, he published in 1859 On the Origin of Species. Okay, and that affected biology, psychology, anthropology, the arts, and literature. It affected all life as we know it, you know, in terms of how we looked at it. Now, Darwin's travels allowed him to document evidence for it. So he described natural selection as a mechanism for evolution. So the mechanism for evolution is natural selection. Okay, as an example, we have these peppered moths. Okay, so theories of Darwinian natural selection and Mendelian genetics and population genetics intersected during the second revolution. Okay, so we have examples of these peppered moths. Okay, so against a dark background, the gray peppered moth would be more apparent. So birds would eat more of these, so there would be more dark moths. And then when the bark was less dark, then you'd have birds eating more of these dark moths, making the gray peppered moths more abundant. Okay, that was actually evolution that people were able to see during their lifetimes. Okay, changes in population genetics over generations. Okay, so Charles Darwin had a very early interest in natural history. He always was interested in biology, in living things. Okay, his father was a medical doctor, a prominent medical doctor, and he was sent to medical school in Edinburgh when he was 16. But he was really repulsed by the barbarity of surgery at the time. People would have to hold somebody down while they were doing an amputation, for example, while the person was conscious. Okay, so he transferred and studied theology at Cambridge, and he graduated in 1831. That was when he went on to the HMS Beagle for his around-the-world voyage to chart the coastlines, especially of South America, and he was actually supposed to be the ship naturalist. Okay, he was assistant naturalist and captain's companion. Here's a picture of the captain's companion. No, I'm sorry, that's actually Charles Lyell's book. Charles Lyell, I think. So Charles Lyell's book is based on John Hutton, that all geological change came from slow processes. Geological changes or earth changes, such as the making of a canyon or the building of a mountain, took thousands of years, hundreds of thousands of years. Okay? So then people were thinking, some scientists were thinking that Earth was very old indeed, because most people at the time thought that the Earth was no more than 4,000 to 10,000 years old, because they went by the list of kings in the Bible. You know, so they thought the Earth was no more than several thousand years old. Okay? But some scientists at the time were thinking that Earth was very old indeed. Okay? We had accumulation of changes. Okay? Accumulated small changes led to big changes. Okay? So Charles Darwin collected plants and animals in South America, the Galapagos Islands, Australia, New Zealand. He kept a daily journal, which he published, and he sent back his journal to England. And when he returned to England in 1836, he was already famous, but he kind of got married. He worked on his collections and his journal. He retired to the countryside. He corresponded with biologists, because biologists do not work alone. Okay, everybody, whoever makes any kind of discovery, builds their work on other people's work. Okay, and they confer with their colleagues, they talk to people, and they write people, or they email people. Okay, so he investigated pollination. He investigated the effects of earthworms and the biology of earthworms. He investigated biogeography, or why living things lived where they lived at the time they lived there. Okay, and he was guided by an essay on human populations and food that was published in 1798 by Thomas Malthus. That's Thomas Malthus. Okay, so that essay actually talked about populations grow, and food supplies and disease and wars limit human populations. Okay, but populations have tendencies to grow exponentially. So if it weren't for not enough food supplies or massive pandemics or wars, then we'd have unlimited population growth, mass starvations. Okay, so then we have wild organisms that are vulnerable to selection in nature. Okay, so Alfred Russel Wallace sent an essay in 1858 to Charles Darwin, and here's a picture of Alfred Russel Wallace. Okay, and he said that natural selection led to the origin of new species. He came up with the exact same idea that Darwin had. Okay, so Darwin and Wallace had a joint presentation to the Linnean Society in London. So the Linnean Society was a very prestigious scientific society. Okay, and in 1859, Charles Darwin published On the Origin of Species by means of natural selection. The evidence for evolution include structure and relationship of proteins, DNA, and other macromolecules. Okay, the fact that the use of ATP as an energy source is universal in all organisms. Okay, cytochrome C oxidase occurs universally. That's an enzyme. Okay, and also fossils. The fossils are simpler in the oldest layers on the planet, and they get more complex in the youngest layers. Okay, so this is some evidence that can be used for evolution, because evolution not only says that some organism evolved from other organisms, but evolutionary theory actually also holds that all living things actually descended from a common ancestor. Okay? Also biogeography, that you have a geographical distribution of organisms, like we saw when we were talking about food. Okay, so microevolution is evolution within a species. So you have natural selection, and the parts of natural selection that lead to natural selection is overproduction. There are more young produced and can survive. Like a flowering tree will produce many more seeds than will ever germinate, and there are only a few that ever germinate, ever grow to full size and reproduce. Okay, there's a struggle for resistance, a competition between individuals for scarce resources. Okay, and for those who do survive and reproduce, they have inheritance and accumulation of favorable variations. So what you have is survival and reproduction of the fittest. Okay, that's what natural selection is all about. Okay, so you can have migration, which allows for a gene flow between populations. Okay, we can also have in microevolution genetic drift. So genetic drift means that the genetic makeup changes due to random events, such as the founder effect. Okay, so if these red and blue dots were a population, or red squares and blue dots were a population of a population, okay, just a population of individuals of different types, and then some migrated to other places. One place might have only red squares, some might have a combination of red squares and blue dots, and some might have only blue dots. So that's actually a random event. What actually gets where is due to chance effects. Okay, so that's called the founder effect. Okay, we also have random chance, having an effect like a tidal wave coming and taking a bunch of animals away somewhere and drowning them. So that's a random chance event that can actually lead to genetic drift. And we have the bottleneck effect, okay, where you have population of a certain size and you have some kind of bottlenecking effect that really decreases the amount of the population. Okay, then you can have recovery in some cases, but a bottleneck effect is also a form of genetic drift. Okay, that will that is a genetic makeup changes due to random events. So these are the three types of events: the founder effect, random chance, and the bottleneck effect. Okay, now rates of evolution. Okay, include gradualism, where change happens slowly and gradually over a long period of time. Okay, like this is showing gradualism. Then you have punctuated equilibrium, where things remain the same for a long time and then they change very swiftly. Okay, over a short period of time, then remain the same a long time. Punctuated equilibrium. Okay. Know when species evolve. That's called macroevolution. Okay, how species evolve. Okay. In order for a new species to evolve, you need some kind of isolation barrier between two populations. Okay, so you can have prezygotic barriers that happen before a zygote is formed. So some of these include a geographical isolation. So an example of that are the red buds or Cirsus species, the eastern and western red buds. You have the eastern red buds isolated from the western red buds, so they cannot hybridize with each other. That actually form two different species. Okay. Okay, then we can have ecological isolation, such as Dutchman's breeches and squirrel corn. Okay, they look very similar to each other, but they live in different kinds of ecologies and different ecological niches. We have mechanical isolation, such as catasetum orchids. Okay, artificial hybrids are easier to make, but actually no natural hybrids are known, because they're mechanically isolated because the insect that fits into a particular flower and allows it to actually hybridize does not allow it. Okay, you have pollinia attached to the insect's head in one case, to the insect's back in another case, to the insect's abdomen in a third case, and in the left front leg in a fourth case. So there's no natural hybrids known of catasetums, even though catasetums tend to fling out their pollinia, if they're triggered by an insect. Okay, because each species will actually attach their pollen or their pollen sacs to different parts of an insect's body. Okay. Now the role of polyploidy. Remember we talked about ploidy a little earlier, about haploid and diploid, triploids and tetraploids. Now almost all angiosperms are polyploids. Okay, it's much more common in plants than in animals. Okay, so how can it actually happen? You can have a diploid hybrid, okay, one set of chromosomes from mom, one set from dad, okay? And you have meiosis, and chromosomes may not pair, so then you can have a sterile plant, okay? Okay, now Darwin's theory of evolution. Okay, it was a great controversy in his lifetime. Okay, so one of the things to remember about faith and science is that science deals with facts and evidence that can be measured or tested, so with empirical evidence, whereas beliefs and faiths are outside the realm of science to disprove. So scientists really should not actually be talking about faith to people. That's my opinion and other people's opinion. Okay. Now scientific naming. Okay. In English you have the scivula or half flower. It's also known as Scivula sericea. Okay. Or they're also known as naupakakahakai. Also the naupakakai, the huahekili. Okay. In Niihau is called the alpaca. In Society Islands it's called Ngau. In the Cook Islands it's called Ngahupa. In Tonga it's called Toi Toi. In Samoa it's called Veveda. And in Guam it's called Nanasu. But there's only one correct name per species in scientific naming. Okay? All right? So the scientific name of that plant is Scivula sericea, okay? Also known as a naupakakai, okay? And the other names that you saw, all right? So there's only one correct name per species. Or you could have one common name for several species, okay? For example, we have mock orange, okay? We know mock orange as a particular plant in Hawaii, okay? But in other places, it could be this plant here, which is a Philadelphia species. Or you could have this Osage orange, which is also known as Maclura pomifera. Or the one that we know, Murraya paniculata, our mock orange that we have in Hawaii. Okay? So without the scientific name, there could be confusion, whether it's one plant with many different names or one name with several different plants having the same common name. So scientific naming is important. Okay, and of course the person who started scientific naming was Linnaeus, also known as Carl von Linné. Okay, he was a professor of botany and medicine at Uppsala University. And in 1753 he published Species Plantarum, and he was the first to assign binomial, okay, a two-name scientific name to a plant. Okay. Such as Mentha spicata was the name that he gave to spearmint. Okay, that's S in spicata should be lowercase. Okay, so he grouped plants by the number of stamens. Okay, and that was an artificial system. He called it the sexual system. It was quite risque, you know. So he'd say things like 10 husbands and 1 wife in a single bed, if he had 10 stamens and 1 pistil in 1 flower. Okay, so he was kind of a character, actually. Okay. Now we have species concepts. Okay, so we have the morphological species concepts, which is based on growth forms. Okay. So a drawback on this is that it can be subjective. The traits may be unclear or inconsistently used, but still some people do use the morphological species concept, which is based on growth forms, on form. Okay. Then you have the interbreeding species concept, which is based on breeding behavior, where a species in a population is able to interbreed and reproductively isolated from other groups. Okay, has to do with mating behavior. Okay, so you can intercross orchid species, but in the wild they maybe do not. So here are different types of Cattleya alliance members. That looks like a Laelia. This looks like a Cattleya. This looks like another Cattleya. So orchids, you can intercross species artificially, but in the wild, maybe not so much so. Okay, that's an example of the interbreeding species concept. Then we have the ecological species concept, which is based on related individuals occupying an ecological niche. Okay, so you can have oaks in a swamp area, or you could have oaks in a wooded area. Okay. We also have the phylogenetic species concept, which is based on evolutionary similarities. Okay, and how things are related to each other. And then we have the eclectic species concept. Okay, eclectic species concept, where they feel that all criteria must be considered. Okay, anyway, so that's all we have for you today. That was a lot that we had to cover. Okay, I hope that you're still awake. I know that I am. Anyway, wishing you best for your next exam. Hope you can make use of this little study session. Okay, and we'll see you next time. Aloha.